Hello students, today we are going to discuss in detail on animal cell culture. What is animal cell culture means? How the cells are grown. Animal cells are grown outside the body, that is in vitro environment, that is outside the living organism, by providing the appropriate nutrients as well as growth condition. So the process of growing cells in the laboratory condition is called as cell culture, that is in vitro we call it as. When it grows in the body we call it as in vivo. So a lot of things we require in the case of the growth and division of the cells, as we have to create an environment which is looking similar to that as it is comfortable in the case of the body of the animal as well as human being. So cell culture is a homogeneous population of cells. Homogeneous means when the cells divides in vitro during the cell culture, all the cells will be looking similar in nature. We call it as clone. Clone means all are xerox copies of cells that will be present and they are genetically identical in nature. The normal growth rate of the animal cells is relatively slow, but usually it requires 18 to 24 hours to divide. This makes an animal cell culture more vulnerable for contamination when compared to bacteria, because bacteria divides for every 30 minutes. So it is easy for them to grow when compared to the animal cell culture. There are essential requirements for the development of optimal growth of the cells, which is regulating the cell growth, that is temperature, proper substrate for and attachment of cells on the appropriate growth medium, and incubator to maintain the correct pH and defined osmolality. All these are necessary for the regulation of the cell proliferation, differentiation, and for the production of the cell. Normally, this cell culture analysis is widely utilized in molecular genetics and immunological analysis, and is involved in surgery and bioengineering and pharmaceutical industry. Here is the history of animal cell culture. It is a routine laboratory technique in 1950 after George Gray has established first human cell line, that is HeLa, from the cervical cancer of the patient Henrietta Lacks. From her name, the first two letters are taken and it is called as HeLa cell line, and it is an immortal cell line that is utilized for the purpose of testing cancerous cells and for the production of the viral vaccines. The cell culture technology has been used in various areas for the efficiency and toxicity of the new drugs what it is developed, and it is involved in the manufacture of vaccines, biopharmaceuticals, etc. With the development of the cell culture technology, a variety of cell culture media have been designed. So wherever we are using means we are using for the development of newer, that is to test the toxicity of the newer drugs. So directly when a newer drug is developed, as such it cannot be given to a patient. So it has to undergo the testing in animal and followed by toxicity testing. So far when we consume a drug, whether it affects our system or not, that we have to identify. That is a minimum lethal dosage LD50 is analyzed for the sake of our newer drug. Once if it is over, the preclinical study, that is in the animal study, is over, it undergoes for preclinical study. A small population of patient is selected, to them it is supplemented and it is analyzed, and further it is followed up in the second cycle. Then after the third cycle of population testing, then only it is continuously monitored for about years together, 10 to 15 years. If there is no side effects, then the particular drug is approved. If any problem in usage of this drug, then immediately the drug is withdrawn from the market. Using cell culture technology, we develop various growth factors such as nerve growth factor, epidermal growth factor, insulin-like growth factor, fibroblast growth factor, platelet-derived growth factors, and transforming growth factors are discovered one after another, and their addition led to the increased cellular proliferation. And during the year 1976 only, we started using serum free media. That is, in order to provide all these factors, the serum is one of the richest source which provides all these necessary factors for the growth and development of the cell. In order to supplement the various nutrients for the development of the cell in the in vitro outside the body, we have to provide the necessary nutrients for growth and development. There are two types of medias are utilized in the case of the animal cell culture. One is synthetic media, another one is biological media. The biological media is nothing but here serum media, another one is synthetic media. So when we make use of the culture medium, it should contain the supplementation of vitamins, amino acids and glucose for the energy provision, and inorganic salts and serum as a source of growth factor and as well as hormones has to be added. Further, the medium has to be maintained with proper pH and osmolality. The media can be either natural or synthetic or artificial media, which is necessary for the growth and development of the cell culture. So this is supplemented. The media containing the combination of amino acid, salts, sugars, vitamins are supplemented with other nutrients, and they are formulated. There are variety of so extensive number of medias are formulated. That formulas are available just like what we have in our market for various products like Horlicks, Complan, like that. No, it's a growth media. Same manner we have similar to that. These medias are necessary for the growth and development of the cells alone. The recipes, we call it as the recipes, are available. What we have seen in the case of the plant cell culture, different types of culture medias are available, no, with different recipes, that is different combination of nutrients. Same manner here also we have different combination of nutrients are available in the case of the media. So normally the some additional components are also needed in the case of the media, that includes your hormones, growth factors and signaling substances which are not present in the basal media, and serum which is required to help in proliferation and to maintain normal metabolism. First and foremost is our biological media is nothing but serum. Serum is one of the important component of the cell culture media. It is considered to be a good source of amino acid, protein, vitamin, carbohydrate, lipids, hormones and growth factor, etc. The serum provides necessary proteins like albumin, transferrin. What is the role of albumin? That is maintaining the biological fluid. That is, it maintains intracellular and extracellular fluid balance. It maintains. In the case of our system, same manner the albumin is required, and transferrin is necessary for the transport of iron within the cell. And in addition, the serum also supplemented with addition factors that helps the cells to bind to the substratum, that is the culture vessel, before they start to divide. The fetal and calf serum is mainly utilized for this purpose. Historical aspects of the animal and tissue culture. Here the various breakthrough in the case of our animal tissue cultures are shown here. From 1882 to 1975, it is shown here. So various types of media we have seen. Natural media, it contains natural biological substances such as plasma and serum. Embryo extracts are available, and types coagulants and clots, tissue extracts, biological fluids. and these coagulants or clots are plasma separated from the heparinized blood and serum and fibrinogen. And tissue extracts are nothing but extracts of chicken embryo, liver, spleen and bone marrow are utilized. In biological fluids like plasma, serum, lymph, amniotic fluid and pleural fluids are utilized for the growth and development in the natural media. What is the greatest? This is the great. It provides necessary nutrient, just like as it is present in the case of the natural environment. But only disadvantage in the case of the natural media is poor reproducibility and reduced uniformity in the case of the cell division. But the exact composition of the natural media are not known clearly for us. Synthetic media or the artificial media is composed of a basal medium such as medium and supplements such as serum and growth factors and hormones are further added. The serum containing media is in which human bovine serum or equine serum is used as a supplement. The quality of the serum varies from batch to batch and deteriorates within 1 year. Therefore, every batch of the serum needs fresh testing for our purpose. Whereas serum-free media are crude proteins factors that as bovine serum albumin, alpha-beta globulins are used as supplement. But it has the ability to make a medium selective for a particular cell type since each cell appears to require a different recipe. And xeno-free media. Xeno means human source components such as human serum albumin are used as supplements and animal components are not allowed in that process. Protein-free media, here in which the peptide fractions are undefined components such as the peptide fractions are used as supplement for this medium. And chemically defined medium in which phosphate buffers PBS and Delbeco phosphate buffer, saline DPBS, Hank's balanced salt solution HBBS and early balanced salt solution EBSS, all these are utilized. This balanced salt solution is composed of inorganic salts as we seen in the case of the plant tissue culture. Here also a mixture of the various inorganic salts are added and it is maintained physiologically with the normal pH of pH 7 as well as that plays an important role in maintaining the membrane potential during the cell division. And there are complex medias are available in the system. That is 4 different types of complex medias are available. Eagle's minimum essential medium, Delbeco's minimal essential medium, Roswell Park Memorial Institute medium and Ham's nutrient mixtures are available. These complex medias are working as cofactors in enzyme reactions and in cell attachment. Sodium, potassium, calcium, chloride, sulfate, phosphate and bicarbonate and inorganic molecules are involved in this media. In order to maintain the osmolality, these medias are utilized and a concentration of sodium chloride is also added and adjusted. Bovine serum are commonly utilized to support the growth of the cell culture. However, there are certain disadvantages in the case of the serum medium. The serum contains insufficient amount of cell-specific growth factors and cannot be used standalone for culture. So it may contain some cytotoxic compounds, some growth inhibitors will also cause inhibition of the cell culture and its proliferation. The serum may also contain some high risk of contaminants like some viruses, fungi and mycoplasma. That is also a problem that spoil the cell culture. The serum in the culture media also interferes with the purification, isolation of cells and products and pharmaceutical compounds. So it is very difficult when I make use of the serum media. Antibiotics, even though it is not essential for growth of the cells, but these antibiotics such as penicillin, streptomycin is commonly used in cell culture to prevent the contamination or the growth of the bacteria and fungi. So these are all the types of media we have seen so far. That is, various types is natural media, synthetic or artificial media, and in the artificial media we come across serum containing media, serum free media, xeno free media, protein free media, and chemically defined media, as well as complex media. Let us see what are all the physical environment which is necessary for the growth and culture of the animal cells. The in vitro, the animal cells is involved in it requires a micro environment, that is nutritional factors, physical and hormonal conditions to enable the optimal growth of the cells. In order to control the temperature and osmolality, pH, gaseous environment, a supporting surface is provided for the cells for its chemical, physical, and mechanical stress. So normally the temperature is provided by the carbon dioxide incubators that are used to grow mammalian cells at 37 degrees centigrade. The cell culture always maintained at 37 degrees centigrade, that is a normal physiological condition, and it stimulates the body temperature of Homo sapiens, that is human being. Moreover, the most of the cells have been derived from warm-blooded animals that will grow optimally only at this temperature. Osmolality. So normally the osmolality of the medium has substantial effect on the cell growth and function. It maintains the integrity of the cell membrane. When it alters, when the concentration of the electrolytes or the salts when alters, it affects the integrity of the membrane, so automatically it resulting in rupture of the cell, or it will swell or shrink in size. So to maintain the osmolality is very, very important in the formulation of the media. Both glucose and salts are the major donors for the osmolality of the medium, and some amino acids too contribute significantly. So generally all the commercial medias are formulated in such a way to have a fine osmolality of approximately 300 milliosmoles. So don't forget, it requires 300 milliosmoles, that is maintained by your amino acids as well as in the case of the salts. So normally the osmolality of the media is measured by the osmometer. And further, during the metabolism, when the cell is metabolizing, it releases the components. It may be acids or alkali that has to be buffered by the system. So in order to maintain the proper pH, so neutral pH of 7 or physiological pH of 7.4, it has to be added with buffer. The buffer is a combination of acid as well as base. So normally we have the natural buffering media which is available in our system is nothing